Good morning, AP Bio. So we are kind of getting towards the end of this genetics section, and I was like, didn't know how much I wanted to get into eukaryotic stuff. It's much more complex than prokaryotes, and we don't know as much either about eukaryotic gene control. However, I did want to just briefly mention a few things. So I cut down this slideshow. It only has a couple slides because I just want to present a little bit on epigenetics next week because I find it so fascinating. And just understanding a little bit will be beneficial. And I'll just kind of give you a broad stroke of what's involved in eukaryotic gene control. So here we go. All right, so let's get started. All right, so what are we thinking about with eukaryotic gene control? Well, just like in prokaryotes, how are those genes turned on and off? What happens? They're not regulated in quite the same way because we don't have like operons. And then we are not a single-celled organism. So different cells are going to need to regulate different genes differently. A big part of that, hence all the little babies, is differentiation of cells, right? So once your heart cell becomes a heart cell, it will always become a heart cell and divide into other heart cells. It's never going to become a liver cell because particular genes are turned on and off permanently because of their specialized functions, okay, which is very different than the prokaryote. All right, so a little refresher on prokaryotes. Okay, what are they? We know this. They're single-celled. They grow and divide quickly. They're really going for quantity, not quality, but they are in their environment. They need to respond to what's around them. You know, all of a sudden, going back to the lac operon, there's a ton of lactose around them. Well, that's the source they have for food, so they need to break that down. That goes away, and all of a sudden, sucrose is all around them. Well, now they need to be able to digest that. Okay, so because of that, genes need to be turned on and off super rapidly to respond to the environment. Okay, where eukaryotes don't need to be that quick typically. All right, eukaryotes, on the other hand, like I was saying earlier, we are multicellular typically, not all of us, not all eukaryotes, but we are. And so differentiation is a big part of eukaryotic gene control, making the different cells that the body needs to function as a whole. Okay. We have a pretty advanced homeostasis or internal balance. So it's not like your cells are going to all of a sudden be all of them bathed in lactose, you know? Yeah, sure, that might go through your digestive tract and you'll have specific cells to break that down, but your heart cell's never going to need to adapt to that. All right. And so because we keep this internal balance, we're kind of like the same things are typically delivered to the same cells. We keep the same temp, all those things. We don't need to respond to the environment as quickly. So instead of using our energy to respond quickly to the environment, we use our energy to regulate specialization so that we can function as this whole organism, serving the body, like it says at the bottom, instead of the individual cell needs. Okay, so how can eukaryotic gene expression be controlled? Well, it can basically be controlled at any level, all the way from the DNA through transcription, translation, the whole protein synthesis, and all the way to degrading the proteins more rapidly at the end of that. Okay, so we are not going to talk about all these today. I just picked a couple that I wanted to discuss, mostly related to the unpacking of the DNA, because that is the portion where we are differentiating a cell and turning certain parts of the DNA on or off permanently. And that's a big difference between prokaryotes, so that's the part I wanted to accentuate. It is also what we call, focus a little bit on the epigenetics, beyond, epigenetics means beyond the genome. So if you're adding things to the DNA to permanently shut them on or turn them off, I think I said shut them on, which doesn't make sense. Sorry, permanently shut them off or turn them on, then it's not actually changing the DNA or the expression that it's changing the ability to express that DNA. So that's what we call epigenetics. All right. But we can regulate at transcription at, the mRNA, how it's cut, like from spliceosomes, how it's transported, the length of the polio tail, translation, how well it's translated. We can actually put a protein on the end and stop translation. So there's just all these ways, and there's even like special proteins. One I like the name of, well, it's ubiquitin, but it's called the chamber of doom at the end that can degrade proteins really quickly if we want to get rid of them. So there's lots of places that we can regulate our proteins or the expression of our DNA. But we're just going to talk about a few. And first, we just need to refresh on the packing of the DNA. We've talked about this a little bit in the past, but we have this huge amount of DNA, right, for our tiny, tiny nucleus. So we can't have it all just loosely folded. So we've got to condense it. All right. And how that happens is around those histone proteins. And then those are coiled up, and then our DNA is folded, and then those folds are folded. And when we're actually dividing, it will be tightly wrapped in the chromosome. Okay, so you can see those levels of a condensation there. So a little more of a look at the histone protein. So the histones are negatively charged. All right. I'm sorry. Don't pay attention to that. The histones are positively charged. So the negative DNA is attracted to them. Okay, so that's how it will attract them. Then we'll wrap around, and then we'll get a clump of 8 of these all together that we call a nucleosome. Okay, and then those clumps will kind of stack on top of each other and coil. All right, so that's just a little organization as we condense the DNA. Well, as you may be thinking, if that DNA is like super tightly coiled around all the histone proteins, which are all clumped together, that are also tightly wound and folded, it would be pretty hard to go through transcription. RNA would not be able to RNA polymerase would not be able to grab a hold of that very well and get transcription going. Okay, and so when you look at the DNA in a nucleus and it's dyed, you will typically see a darker region, the heterochromatin, you can see there, and that area is tightly coiled. That DNA is probably DNA that you don't need for that cell. It remains tightly coiled and we never really access it. Where the euchromatin is lighter colored, it's not as tightly packed. The RNA polymerase can get at it and we can actually transcribe that DNA. Okay, so heterochromatin is basically genes that are turned off. All right, now those turned off genes, if we're looking at a kidney cell, it will probably want to permanently turn off things for genes that are dealing with the cones in the back of your eye. Okay, so not only will that heterochromatid be tightly coiled, it can also have a methyl group attached to the DNAs, which is basically a permanent shutoff of that DNA. A great example of this that we've talked about is the Barr body in females. That X chromosome is shut down, okay? It's heterochromatin, it's tightly coiled, it has methyl groups all along it, and we're just never going to access it. It's permanently inactivation of that DNA. And so part of differentiation is that occurring. Now, what's interesting with this is certain epigenetic cues, choices, habits, lifestyles can actually cause methylation of certain bits of DNA that could end up being a good or bad thing. And this is where your lifestyle can really affect your genetic expression, which is crazy. All right. And on the flip side of that, things can be loosened. Okay. I'm always nervous when I'm saying this if I'm acetylation, okay, where you have the acetyl group attached to the histones instead of the methyl group. Well, now this loosens and makes the DNA easier to access. So this helps eukaryotic gene expression. All right. Now, those are the biggies I wanted to talk about, okay? Because that's big, like big picture, differentiation, like what cells have access to what DNA. So that's the start of DNA regulation in eukaryotes. There's lots of other steps, so I just included this one last slide so you could overview it. We talked about DNA packing a little bit, but also all sorts of transcription factors can help regulate, and then the enhancer region as well. This is all eukaryotic gene regulation. Then after transcription, we've talked about this in protein synthesis, but mRNA processing, how that particular gene is spliced, will regulate its expression, and also how long the poly A tail is. And then something we didn't talk about, but there are these little tiny RNAs that can actually attach to complementary RNA on the mRNA strip. The SI stands for small interrupting, because basically if it attaches to the other side, it turns it off because now the mRNA can't go through the ribosome. Okay, so that's a quick shutdown if we want to stop transcribing. There are proteins that can attach to the mRNA sequence and stop translation, so the ribosomes won't attach to it. And then there's also different ways that we can increase protein degradation. Sorry. So we have too much of this protein. We don't want it anymore. We're not going to use it, and we'll break it down. Now, obviously, we've already put in all the energy to make those proteins, so this wouldn't be like an ideal way if we didn't want protein synthesis to occur. But if we're like, oh, we don't want those proteins around anymore, we can then break them down, and so we would no longer be expressing that gene. Okay, again, we're not going to go into the details just because of time and this weird end of the year, but I wanted to give you a brief overview of just showing you, hey, there's a lot of ways we can regulate genes in eukaryotes, and we're going to talk just a little bit mostly about the big time regulation of gene expression, the more like permanent turn on and turn off of of genes, and epigenetics for a few days next week. And then we're going to start reviewing. And then we're going to have a big old test over all of genetics, which goes all the way back to mitosis, which is crazy. Okay, so know that that's in your future. Yay. All right. Have a good weekend. See you guys.